Hello, my name is John Powers, and I'm working with Ann Sakai, Steve Weller, and Diane Campbell at the University of California, Irvine to understand how floral scent evolved in an island plant radiation. Today, I will focus on how floral scent varies along the axes of time, space, and sex for a plant that is spread to multiple islands. This work is exciting because it is the first study that we know of to compare scent and genetic divergence in a wind-pollinated plant species. During the evolutionary transition from insect to wind pollination, floral displays that advertise to pollinators such as showy petals, nectar production, and attractive scents are often lost or heavily reduced. However, other volatiles that deter or defend against herbivores may remain if they improve fitness. If scent is not completely vestigial, it is unclear whether the daily cycles in scent that formerly attracted diurnal or nocturnal pollinators will remain in wind-pollinated taxa or be lost. For insect-pollinated plants with separate sexes, males and females must produce similar volatile blends to attract the same pollinator. Wind-pollinated species, which often have separate sexes, do not face this constraint and are expected to have greater sexual dimorphism in scent. Floral scent blends are known to vary between populations, and for insect-pollinated species, this variation is often linked to local pollinator communities that exert different selection pressures and lead to a geographic mosaic of scent. In species that do not rely on insects for pollination, we predict that neutral processes of gene flow and genetic drift would shape geographic variation more than local selection. We tested these predictions in Schiedea globosa, a species endemic to the Hawaiian Islands that grows on rocky coastal cliffs and has several adaptations for wind pollination compared to its moth-pollinated relatives that occupy mesic forest habitat farther inland. The most striking adaptation for wind pollination in Schiedea globosa is the condensed globe-like inflorescences which sway back and forth in the wind. Unlike its moth-pollinated relatives, this species is subdioecious, meaning individuals are female, male, or hermaphroditic. You can see the copious pollen production of the male anthers and the long stigmas of the females that collect pollen grains. Whereas most species in this genus are single island endemics, Schiedea globosa occurs on the windward coasts of 5 Hawaiian islands, 3 of which were part of a contiguous island called Maui Nui for the most of the past million years. We asked the following questions about Schiedea globosa. Are there daily temporal changes in floral scent? Do females and males produce different scents? And does scent divergence increase with genetic distance between populations? We used dynamic headspace sampling and GCMS to characterize the floral scent of 1 to 4 individuals per sex per population during the day and following night. Inflorescences were bagged, allowed to equilibrate for 30 minutes, and then pumped for 30 minutes. We used sequence data for 1 chloroplast and 2 nuclear loci from a previous study by Lisa Wallace to calculate genetic distances between populations and infer a neighbor joining tree of the species. We first examined changes in the total scent output among sexes and over the course of the day. Scent samples taken from the same inflorescence during the day and night are connected with a line. Surprisingly for a wind-pollinated species, we found that floral scent emissions increased 51 to 70% at night. This daily change is similar in magnitude to those of moth-pollinated relatives. Total emissions also varied among sexes, with males producing 64% more scent per flower than females at night. This difference could be partially explained by the larger size of the male flowers. In addition to differences in total scent emissions, there were also differences in the relative emissions of volatiles across sexes and time. To visualize these differences, we conducted a canonical analysis of principal coordinates of scent composition, which is a type of constrained ordination that maximizes the projected variation between groups, in this case, sex and time of day. Day and night samples from the same plant are again connected with a line. Each plant showed a change in scent composition from day to night. Males and females produce different scents on average with some overlapping individuals. Hermaphrodites, in green, produce scents resembling those of either males or females. We can conclude that there were quantitative but not qualitative differences in the scent blends emitted by males and females, and the ratios of the volatiles changed as the total scent emissions increased at night. Now, using the same ordination, we can ask what volatile compounds were responsible for these differences. Compounds on the left increase at night, those on the right increase during the day, and those at the top and bottom are found in higher relative amounts in males and females, respectively. A suite of nitrogen-bearing aldoxenes that smell like old socks, in orange, increase at night and are relatively stronger in males. Many of these nocturnal aldoxenes are known moth attractants in other systems, although none of the nitrogen compounds are produced by two moth-pollinated relatives on Oahu that we have studied intensively. This suggests that the daily cycling of these compounds is not inherited from a moth-pollinated ancestor. Aldoxenes are also induced by herbivore damage in other species, so their function here is unknown. The benzenoid phenylacetaldehyd in blue is a general moth attractant and also increases at night, a feature shared with the moth-pollinated relatives. Terpenes, shown in purple, increase during the day and include linalool, a compound that is known to be either an insect attractant or deterrent depending on the concentration, and DMNT, a leaf wounding volatile that often attracts predators of herbivores as a form of indirect defense. Finally, some aliphatics in green increase at night and are responsible for the butter popcorn smell, while two other aliphatic alcohols discriminate between males and females. To reconstruct a hypothesis for how Schiedea globosa spread across the Hawaiian Islands and determine which populations are closely related, we built a tree based on previously published sequences from the populations we sampled. This revealed strong genetic differentiation of populations between islands, with the exception of two populations from Hawaii Island, which were genetically similar to those on Maui Nui. The phylogenetic pattern was generally consistent with colonizations from older to newer islands, although support of the deep nodes with this data set is poor. The origin of the species, at least of the extant populations, seems to be southeastern Oahu, with other populations clustering together. To examine how scent composition varied within and between these populations, we performed another cap ordination separating populations. We will show the results separately for day and night samples. Here, lines are connecting individuals of the same population. The major split in scent composition separates the two populations from southeast Oahu, Makapu'u and Koko Head, from all other populations, including those farther north on Oahu in ridgeline habitats. This scent difference exists during the day and during the night. During day and night, the southeast Oahu populations produce relatively more DMNT, linalool, Z-isobutylaldoxine, and phenylacetaldehyd. The other populations, which according to the phylogeny diverge early in the history of the species, produce less of those and more 3-methylbutylaldoxine at both times. If the placement of the northern Oahu populations is correct, these differences could have built up between the southeast Oahu populations and the others after they were genetically isolated. To show the relationship between scent divergence and genetic distance, we averaged these metrics by population pairs and performed a Mantel test of a linear correlation between the two for each combination of sex and time of day. There was a statistically significant positive correlation between scent divergence and genetic divergence for males during the day and night in blue, although females in red were more variable and did not show a clear pattern. This result is consistent with our hypothesis for neutral processes driving interpopulation scent differences. In males, closely related populations had more similar scents, and distantly related populations had distinct scents. In summary, despite adaptations for wind pollination, Schiedea globosa produces a strong floral scent reminiscent of butter popcorn and old socks. This scent intensifies at night, but the composition of the nighttime blend does not match moth-pollinated relatives, with the exception of phenylacetaldehyd, a general moth attractant. Males produce more scent for flower than females and show some quantitative differences in scent composition. And finally, scent divergence increases linearly with genetic distance in males, which is consistent with this trait being shaped by neutral evolutionary processes. In the future, we plan to better resolve the phylogeny within this species and with other species in the genus to clarify how many transitions to wind pollination have occurred and what floral volatiles have changed in those transitions. Thank you.